Okay, I'd like to introduce our next speaker, Richard Levenson, who's currently at the University of California, Davis Medical Center, and his talk is on the screen. Richard?
Thank you, Stephen. Excuse me. This is going to be a complete change of pace, if I can get it out from my lungs to you. And what I really want to focus on is trying to capture the implications of not what we're doing now, but what might be coming down in the future in terms of what is required of pathologists in a context of perhaps a misconceived dedication to what's called personalized medicine. And by that I mean specifically what it's being used for. I believe firmly that we should personalize our medicine, but this term seems to mean deep characterization of tumor epithelia. And I think that's been a or it's not likely to work, and I'll give you an analogy why that's going to happen. And then the other point is what if there are new therapies coming along that are completely orthogonal to everything we've done up to date? And I will show you an example, three examples that look extremely promising and really change the paradigm in my mind as to what we're going to be doing and be asked to be doing in the future. The reason this talk is about multiplexing is it's the pathologist's approach to deep molecular characterization. And I'll show you some possibilities where this could be useful and some where it hasn't been that effective. And I'll end with a cat video. So this is just my sort of couple of hate slides on personalized medicine. This is 2006, very complicated pathway analysis. In 2012, we just had a publication in Nature that looked at many different ways of deep molecular characterization of over 100 tumors with RNA, DNA, and the findings were actually somewhat disturbing for those who were hoping that this would provide a kind of roadmap to effective therapy. And I think it's going to be because we are not necessarily looking in the right places. And so one of the themes is that the stroma is where the action is not so much the tumor. The tumor is kind of an emergent event reflecting host susceptibility to cancer, and so the susceptibility is what we should be targeting. And then, as I mentioned before, some of the other things, maybe all of these analyses are relevant. If we really had an effective therapy, we don't need to do genome-wide sequencing. For example, if a patient comes to you with a headache, you don't sequence them, you give them aspirin. So I'm hoping that we're going to move over this complexity hump into a new era of increased simplicity, and that's just a hope, but it's something to think about in terms of what kinds of demands will be placed on us. And just a sort of thought piece here, and no cheating from yesterday, where I gave a talk in the image analysis section. What are those things? Well, Staphylococcus aureus, a dread pathogen which kills people, but 20% of us bear it without any consequence in our nose and oropharynx. And so the consequence, you can't look at that. You could characterize that all day and you still couldn't tell if that was going to cause a problem in a patient. The answer is it's the patient that determines whether this will be a serious infection or not. So back to the deep genome sequencing stuff that we've been looking at. There's a concept of driver mutations, and the hope is if you can find a driver mutation, you can target that. The problem is that 100 tumors were examined. There were driver mutations in at least 40 different ones, and there were 73 different combinations of driver mutations. And the optimist would say, oh, look at all these targets for potential therapy, and the pessimist would say, yes, at $800 million per therapy. So that's problematic. And I liken this characterization approach to fingerprinting. Fingerprinting is extremely personalized. That fingerprint is shared by no one else on Earth, and yet it's also highly uninformative. I couldn't tell you if that's a man or a woman, old or young. So I think we've been perhaps misled by the power of our molecular approaches. and I think we're learning their limitations. So just to touch on the topic that I'm supposed to be talking about of multiplexing, I'll show you how in pathology we can approach this in terms of tumor characterization with the added benefit compared to these a lot of the genomic approaches of course, the spatial context. And so here we have a minimal multiplexing. It's a double immunostain with a counterstain, but in bright field, it's still hard for our human eyes to separate brown and red and blue when they're all overlapped with each other. And so I've circled the normal duct of this breast cancer specimen on the left and the invasive component on the right, and then there's some extra sections there, and the question is, are they normal or cancerous? And you can, using multispectral imaging, or if you don't need it for this few number of dyes, but if you wanted to do 5 or 10 or 15 different, you need to use additional approaches, and there are some potential applications for high multiplexing. And I hope to be exploring that using a variant of mass spectroscopy in the coming year. But in this case, this minimal case, you can find where the ER is expressed and the PR, the red progesterone receptor, and then you can use just Boolean output to show the double-labeled cells, and you can see that the invasive cancer is highly double-labeled, the normal is not, and these extra little bits of brown staining cells are probably normal, and you can tell it morphologically. And that's physiologically natural in women, especially cycling women. The breast goes from ER positivity to PR positivity. It switches, and you rarely have ER and PR simultaneously in a single cell. So this is cancer dysregulation, and the hope from these early characterizations and more complicated ones is that the molecular information that we get would turn out to be meaningful for both prognosis and for therapy selection. So here are 4 images of breast cancer TMA. These are 4 different patients, all stained on the same slide with 3 different immunostains for estrogen receptor, progesterone receptor, and HER2. And actually, an extra component which I talked about yesterday but don't have time to talk about in any length today is that on the left are the raw images, what you would actually see through the microscope. On the right is after a couple of stages of automated image analysis where the cancer regions were detected, the nuclei, individual nuclei were circled, and all automatically, and also beneath the hood here, multispectral imaging has unmixed the 3 molecular signals. And you can take all of that data and put it into scatter plots, and our brain can just about tolerate 3 dimensions. It begins to fail dramatically at 4, and if you think about 15 or 16 or 20 or 100 different dimensions, you're in a world of hurt in terms of trying to see patterns. But in this case, these were the 4 different patients, and each cluster is a cluster of a single patient. You can see just right away that there are 4 different phenotypes. This person down here is ER positive only, no PR. This is HER2 only, and this red cluster here is ER, PR, HER2 combined. So that's a sort of triple positive. And the hope was that molecular phenotypes would suggest targeted therapies, and that's certainly true in terms of estrogen receptor and Herceptin. The problem is that these therapies only last a while, and patients very often find a way around them, and I'll talk about that in a second. So here's the second. So are we off target with targeted therapy? And my analogy has to do with the German tank, the Panzer from World War II. This was and so let's call that cancer. This was a truly fearsome enemy, and so the question was to the English, especially early on, how do you kill a tiger? And the answer was that they could do it, but it took 5 Sherman tanks to take out a single Tiger tank, and if they did that, only one tank would survive, only one Sherman would survive. So perhaps there was a vulnerable pathway in this entity that could be targeted, and it turned out to be ball bearings. So in 1939, Schweinfurt was the city that produced almost all of Germany's ball bearings, and so it made sense to bomb it, and the Allies did 22 times with over 2,200 aircraft. Here's an example of one bombing run. And the result was unsatisfactory. The initial German, of course, it immediately ablated the most of the ball bearing output, but it turns out that Germany had two strategies. One was to buy up the entire output of the Swedish ball bearing industry, and I'd sort of forgotten this, but Sweden was neutral and was shipping stuff to both the Allies and the Germans, which was good business. But it turned out that there were enough ball bearings in the rest of Germany to keep things going, and then the Germans built the factories again underground, and so they became essentially resistant to therapy. So it's a long way around to saying that the targeted therapy usually ends up being leading to resistance, and so you get a temporary respite. So there has to be, if we're really going to succeed, we need to think beyond this box. And so another box, and there are little boxes here, to think about is, are we looking at the wrong box actually in the tissue? This is just normal breast, and a blow-up brings up the two compartments in general terms that are of interest. On the right is the epithelium. This is where we've been focusing to date. It's where the cancer arises or seems to arise. And it has various cell types and it leads to different molecular phenotypes and morphological phenotypes that we recognize. On the left is the stroma, an essentially sort of apparently bland mixture of supporting cells and kind of infrastructure. But it turns out that this is where the action may probably be. You've got the endothelium, the vasculature, which feeds the tumor. You've got lymphatics, which take away waste. You have the collagen, which very importantly has a molecular interaction where you have mechanical signaling to the tumor, and you have, importantly, immune cells. Here are a couple of lymphocytes. And it turns out that current research suggests that the molecular phenotype of the tumor that arises here is far less important in terms of outcome and even therapy than what's going on over here. And I'll try to support that. And of course that has implications for what we look at as pathologists, and I think it's worth talking about. So we heard a talk yesterday from Andy Beck from Stanford. He's now at Beth Israel. Did some very nice work looking at just the morphology of breast cancers with a semi-automated way of extracting features and then looking for the significant, and then correlating these features with outcomes. And he was able to, on hundreds, or he and his colleagues, on many hundreds of breast cancers from published sets, a training set and then test set, were able to show that you could use analysis and segment the outcomes. This is no big deal. You can sort of get that through other means. What was interesting, though, is if you did the analysis, this is what turned out to be not significant. With this extra information, you could then begin to find out that these parameters, the 70-gene prognosis signature, ER positivity, lymph node positivity, molecular phenotypes, ERB2 molecular subtype, these turned out to be non-significant in predicting outcome. What was significant? The score that I showed you based on, I didn't go into any detail, but based on morphological parameters in both the stroma and the epithelium, a hypoxia signature, molecular hypoxia signature, and the age of the patient. And in fact, just 3 stromal features predicted outcome better than 8 features calculated from the epithelial compartment. And this might be a statistical anomaly or a peculiarity of this particular study. But I'll show you in a second what happened some years ago when a similar kind of analysis was done with more molecular precision, using microdissection to separate the gene expression profile from the tumor compartment, or the epithelial compartment, and the stromal compartment. And I just wanted to plug a little bit of work that we've been doing at Davis, which is just to develop machine tools to separate compartments morphologically in tumors that can lead potentially to improved tools for exploration, possibly diagnosis. So this is just one tool where there's sort of fluffy stroma and dark pink stroma, there's cancer epithelium, cytoplasm and nucleus, and so with our tools that can be very easily trained and automatically applied, you can pull out these different compartments. And I'd be happy to answer questions, but I won't discuss this any further. So I mentioned the molecular expression research, which also targeted or turned out to show that the stromal gene expression actually trumped epithelial gene expression. And this is from Morag Park at Michigan in 2008, so it's a long time ago in dog years. And you can see on the bottom right, that these signatures very well separated good outcome from bad outcome. And remember, this is from the stroma, not from the tumor. And in fact, and very reminiscent of what I just showed you from Andy, the outcome was independent of ER, independent of HER2, independent of lymph node status, age, grade, and tumor size. I mean, this is really profound, profound insights into what's going on in our cancers. And I think people are kind of avoiding this intellectually because it doesn't fit with how we've been thinking for so many years. So that was my first theme. I have two more themes. The second theme is, let's move on and wonder what we would do, as well as what the medical field would do, if we had drugs that worked actually on everything. What if cancer is really kind of one disease and not 200 diseases? What if we discovered or stumbled on drugs that actually worked on many cancers? That would make our lives much easier. We just have to say cancer, right? No more phenotyping. Well, I'll give you two examples. One is from a company called Novellos, and actually it's taking advantage of the expression of lipid or changes in lipid rafts in tumor cells, and a phosphoether backbone preferentially binds to 52 out of 54 animal models of cancer, and it's also where it's in clinical trials in humans. It has some really remarkable attributes which I don't have time to go into, but it looks like a very powerful platform. You can see there's a I-124 on there. You can put different I-125, I-131. You can put fluors on it. You can use it in many different ways. Here's a fluorescent version of it, and you can see on the bottom left there's skin in that little white box, skin on the left, and then melanoma on the right. This is uptake of this fluorescinated phosphoether. Here's a bunch of cell lines, and you notice it goes into the cytoplasm and strictly avoids the nucleus. And that will become relevant in a second. And it has some really tremendous benefits over as an imaging tool. If you look at the left on this mouse model bearing two prostate tumors on left and right of the flank, as you know, FDG, fluorodeoxyglucose, is used to detect cancers, but it has a big drawback. It is very sensitive and very sensitive to inflammation. So there was induced inflammation that says I up there, and then H is the heart. And the uptake in the tumor is fairly limited. On the right, same rat. Same prostate, same rat, injected with the I-124 labeled phosphoether, and you can see that the inflammation in the heart don't show up, and there's intense labeling in the flanks. And if you look down at the bottom, they're just examples of adrenal, pancreas, brain, bladder, prostate, colon. It seems to be a semi-pan tumor targeting agent that is not only for imaging but also for therapy. Here is an iodinated injection into a mouse with tumor in the upper right area here, as you can see, over several days, and it persists in tumors for days to weeks, which is, I think, in a good case. And you can see by 9 days that tumor has disappeared. This is from a single injection of this agent. And I don't have time, but there are studies in mice showing good survival in a variety of lines. So again, pan-tumor, you can use it for imaging, you can use it for therapy, you can use it for follow-up. What does that mean to us as pathologists? Not now, but 2, 3, 4, 5 years from now if it gets approved. It also is very effective for intraoperative guidance. So here's a tumor, xenograft, I can't remember which one. And 24 hours after injection, it's lighting up only the tumor. You can take a flap and examine using, I think, a Canadian device for intraoperative imaging, and you can actually make sure that your resection is adequate. And there are very few tumors that can resist a complete excision. So this is a very promising development. There are many different approaches in this area, but this looks like a nice one. So that's one example of a pan-tumor therapy. I'm going to give you another example that's also in moving along, and this is an extremely baffling complex from human breast milk, discovered by chance by a microbiologist who was looking to see what about milk might prevent bacteria from binding to epithelial cells. And she was using HeLa cells as the model, and it turned out that one fraction completely killed all of the HeLa cells and had no effect on normal cells. And it causes apoptosis, and unlike the previous one, you can fluorescate this and show that it goes into the nucleus and then causes apoptosis. You can see the electron micrograph on the right. The cell on the right is apoptosing. The one on the left is controlled and perfectly happy. You can, as I said, you can make it fluorescent. Unlike the previous agent, this one goes to the nucleus and causes apoptosis in, interestingly, a BCL2 independent fashion. The mechanism of transport is not known. It's an exogenous protein lipid complex that reaches the nucleus. It's going to be some fascinating biology. On the bottom is colon cancer model, I believe. So it's one piece of tissue, and you can see that on the left is normal, on the right is the cancer, and you can see Hamlet has gone to the cancer. And it is intrinsically cytotoxic. And you can also use it for imaging in vivo. Here it's detecting on the right, on the left are appropriate controls. On the right, it's fluorescinated Hamlet is detecting bladder cancer in a live rat. So one of the things that I'm hoping to do in the coming year is to look at this and try to correlate Hamlet binding with known clinical outcomes and other patient parameters. The biology is not known, but we at Davis have some wonderful mouse models of developmental breast cancer. So we can see where from normal to cancer the binding starts. And actually there's some very interesting work relating this to c-Myc expression and intermediate metabolism. So finally, and I promise you this has a cat video, the third thing that also affects how we as pathologists will interact with cancer is the question of immunoprofiling and immunotherapy. And this also has something you can't really say about chemotherapy these days. This has potential for cure. And so there was a recent headline in Medscape saying Immunoscore could be, quote, transformational in oncology. And the Immunoscore is sort of shown here. There are various sort of parameters they're trying to figure out what the best way of detecting the immune response to cancer is. But it turns out to be highly prognostically significant. And that goes back to my stress on the theme of stroma, because these are stromal cells, they're not tumor cells. They reflect the host response. And here we have 6 patients, and we're looking at CD3 positive cells, so T cells, in a liver metastasis. So you can see liver and met on each thing. And then there's low, medium, and high, or low, high, and high levels of immuno incursion. And you can see the survival goes from 3 months to 11 months to 21 months just looking at the T cells. And this is metastatic disease. So this is, I think, a very profound finding. And then I have a friend at City of Hope, Peter Lee, who did some work before at Stanford and then now continuing at City of Hope, looking at the immune response in lymph nodes next to tumors, so not in the tumor at all, looking at different immune cells, how they cluster, where they go. Here's more multispectral imaging. So this is an example of metastatic tumor in a lymph node with various responsive cells, and if you want to, you can unmix and show the different species. But if you do quantitation, this is old data. He wasn't able to send me his most latest because my email didn't work. But if you look at just CD4 density and CD1A cell density in the lymph nodes, you can separate outcomes dramatically. So again, this is better than what we get by looking at the cancer itself. So Finally, talk about so that's immunoprognosis. What about immunotherapy? And this is a very, very wide and deep field about which I know moderately little. But I'll show you one example that struck very close to home. I was very intrigued by a company called Immunophotonics. I'm unpaid and unconflicted advisor to them. But what they use is laser ablation / immune stimulation plus an adjuvant, glycated chitosan. And you can see that the various groups of mice did poorly, and the line on the top is not the top of the graph. That's actually the survival curve of the mice that were treated with laser and adjuvant appropriately. And like that. So that's therapy. And in fact, it works in patients in some sort of anecdotal series. Here you can see on the top, basically left and right, you can see the arrow pointing to lung mets in patients with stage 4 breast cancer in Peru before and after therapy, and you can see the lung mets disappearing. Those were not treated, just the recurrent tumor deposits in the chest wall were treated, but there was a systemic immune response that was generated, knocking out the mets. And here's just an example of what happens when you fire up the immune system. There's a macrophage going after one of the cancer cells. I don't think it tells you anything, but it's dramatic. Now, finally, this is what I promised you. Here's our cat, Gabriella, and an absolute sweetie. Unfortunately, she was a heavy smoker. The first 3 years, I'm not kidding, first 3 years of her life, she was with a homeless woman living in a shelter in a one-room, and the woman smoked 3 packs a day, and so she had no place to go. So she got a lot of secondhand smoke. And we noticed her sneezing and sort of behaving funny last November. Turned out she had invasive squamous cell carcinoma. And here's a low power, medium power, and high power of these invasive cells in her gingiva because it was coming out around her teeth. She received an operation. This is before the operation. That's an X-ray, and you can see there's sort of lucency on the right. That's the left. And then after the operation, here's a CT scan. You can see she's lost a lot of teeth, right? There's only 1 incisor, not 2. And the arrow is pointing to an area where there's actual bone loss. And so and this is a terrible disease. There's no treatment in cats, 10% 1-year survival. So I called my friends up at Immunophotonics and said, what can you do? And they said, well, fortunately for you, we have a clinic in Arkansas that's just starting, and we were just moving from Boston to Davis, and we detoured through Arkansas, stopped, and got therapy 1, and we got 3 therapies total of the Immunophotonics. And so here is the laser in a sort of fiber optic probe, glowing red. And then here's the poor cat getting the laser up through one of the holes where the teeth were. And the syringe down there is the adjuvant that will be injected subsequent to the laser therapy. And that's a thermometer up there, a video thermometer, making sure that the thing isn't being overheated. And this was in January. And so this is 8 months after diagnosis and after 3 treatments. And she does this, and then she gets distracted. So I yell at her and say, come on, kitty.
Come on, kitty.
See? Come on, kitty. And then she wakes up again and does that. And then she looks at me and says, was that okay? So I consider this, it's a cohort of 1 or a cat horde of 1. It's promising. And so that's an example of immunotherapy, and my question to you, and I will come back in my summary slide after this, but I think digital pathology has a great role to play for all of the reasons that Sylvia said, plus the advanced tools that we can bring to bear, including image analysis and quantitative, spatial, and molecular comprehension. But we need to think about where the biology and where the oncology is going. And this is way in advance of what oncologists are thinking about yet. I mean, this is fringe stuff I'm showing you. But it still is worth thinking about. So I suggest that there are new tumor-specific vulnerabilities that might be pan-tumor-specific, as well as host responses will help determine the ultimate outcome. And the proximate players are inflammation, immunity, and metabolism, so perhaps we should be targeting our probes in those areas. And I think novel therapeutics will redefine prognostic and predictive metrics. And so my conclusion is digital pathology tools should reflect these insights. Thank you very much.
Cat videos? So Richard, I'll ask.
Please.
What you're talking about is the fact that we're trained as pathologists to look at tumors. That's our job. We're a pattern recognizing optical cortex system. And that ultimately, in some ways, we may be defeating ourselves, and we may not have the skill set to find the other metrics that may be more important in the prognosis and prediction of response for patients?
No, we have the skill sets, but we need to follow the biology, even and in advance of the mainstream pathologists, oncologists, even biologists, because we use the tools we have, but there's nothing magical about looking at an immune phenotype or looking at stromal morphology, but we simply don't. Because we don't know how to or what to look for. I'm saying we should, and part of our role is not just to bring in novel cameras, important as they are for the practice of pathology, but actually to advance the science and the art.
Since you can, from Attila Tarnock's work, look for about 18 different antigens on lymphocytes, have you looked at any cell lymphocyte subsets to get an idea of who is leading the pack there?
Well, I have helped Peter Lee, but that's his goal is at City of Hope to drill down into the immunophenotype to figure out who's doing what and who shouldn't be there.
So of the stromal signatures that you're getting, since lymphocytes or immunocytes are certainly present and presumably are active, is the stromal signature just another way of looking at tumor immunology, or are there signatures that are coming from the non-immunocyte stromal cells that are important?
It's a stroma is a very, very complicated. So there are the immunosignatures. There's also the cytokine-chemokine crosstalk. There's also the talk from the tumor to suppress the immune system, because that's what they do. And so there is a lot going on in the tumor that is affecting the stroma, and so those are relevant signatures. I didn't mention it, but I just saw yesterday that the action can be distilled even far away from the tumor. There was just a couple of papers in Lancet this week showing that you could get tremendous prognostic significance looking at genes in circulating white blood cells in the blood reflecting the immunorepertoire that's been energized or not by the tumor. So we don't actually, and so another theme is potentially as we understand more and more, we may need to do less and less. And maybe just a blood sample will be enough to pull out meaningful signatures.
Lovely talk. Congratulations. And I think it does come back to say that, the molecular genetics is not going to usurp morphology. You're right on bringing all of these other out-of-the-box things we should be looking at. But I do think we also need to think about inherited or germline alterations that may also play a role in the responses. And you just said that with your last sentence.
Oh,
so
okay.
Thanks.
Thank you.
Thank you, Richard. So we're on a break now. It is 10:22. We're going to reconvene at 11:20, and you can watch me be provocative, I guess, is the most polite way of saying it. I guarantee you it's going to be provocative.